Here in part 4, we'll talk about the dark reactions of photosynthesis, the means by which the energy captured in ATP and NADPH is transferred to carbon-carbon bonds. Remember from the electron tower, if we can produce molecules such as glucose, we've stored a lot of the chemical energy in those carbon-carbon bonds, which when then metabolized with oxygen to produce CO2 and water, results in a large energy drop, lots of energy being made available for biosynthetic reactions. It turns out that when we begin to look at the carbon dioxide fixation pathways or photosynthesis in purple bacteria and green sulfur bacteria, they're different. In purple bacteria, a 5-carbon and a 1-carbon are combined to form 2 3-carbon molecules known as the Calvin-Benson cycle, or the C3 cycle, because the first stable bonds, chemicals that are produced, contain 3 carbons. It turns out that green sulfur bacteria have a different pathway, the reverse TCA cycle, reverse tricarboxylic acid cycle, basically running the anaerobic respiration backwards. Here, a 4-carbon molecule combines with CO2 to form a 5, and the 5 and the 1 then is combined to form a 6. That is a very functioning photosynthetic cycle, as well as the Calvin-Benson. It turns out that during the course of evolution, cyanobacteria adopted the Calvin-Benson cycle, not the green sulfur cycle. It turns out that that pathway has a downside to it. Here you see the Calvin-Benson cycle, in which we have carbon dioxide combining with a 5-carbon molecule, ribulose bisphosphate, through the action of an enzyme called rubisco, ribulose bisphosphate carboxylase oxygenase. This pathway was adopted by cyanobacteria at a time when there was no oxygen in the environment. We can take a 5 and a 1 and produce 2 3-carbon molecules that are then used either to generate larger molecules or other metabolic pathways. It turns out, though, that rubisco is also sensitive to oxygen, so that as cyanobacteria were increasing the oxygen concentrations, they were decreasing the efficiency of this enzyme, because the active site for rubisco to take up a carbon dioxide molecule is the same active site to take up oxygen. Far less sensitive to take up oxygen, but it could take up oxygen, and this would ultimately have an effect on life on Earth when CO2 concentrations became low. So once again, when we begin to look at purple and green bacteria, what we find is that cyanobacteria adopted both photosystems, but they adopted the carbon dioxide fixation pathway associated with the purple bacteria. So, first clicker question: what aspect of photosynthesis did cyanobacteria adopt from purple bacteria? The answer is C, both the photosynthetic reaction center and the CO2 reduction pathway. Second question: what aspects of photosynthesis did cyanobacteria adopt from the green sulfur bacteria? And it's A, the photosynthetic reaction centers, not the reduction pathway. Based on what we've talked about now, so far, you should be able to answer these questions here as part of your study guide.